Right, thank you to the SCS for inviting me to have this flash talk. I'm going to tell you about ENQUIRE, software to annotate my biomedical literature of interest. To make it more concrete, I came up with basically an example of a day in the life of a systems biology like me. For example, at 9 a.m., a fellow researcher typically would enter the office with some new ideas. We're going to cure melanoma, they might say, because they found a study in 2023 about immunotherapy design that can impact the tumor physiology. And we just need a network for immunotherapy resistance in melanoma. And then we set some database up, do some RNA-seq analysis, train some AI models, we identify poorly characterized genes, druggable targets, we publish, we're getting famous, they might say. And then after 15 minutes, they just pledge, just to kickstart this amazing project, can you build a context-specific network for me real quick? And of course I say, yeah, sure. But I have no clue, I realize 30 minutes after, what causes immunotherapy resistance in melanoma. Then I start searching the literature and I find thousands of results, no matter the tool I use. The best I can do is with mesh terms to fine-tune my query to 100 papers. So, okay, I start motivated, and after 1 hour, this is more or less the state I'm in if I try to read all the 100 papers myself. And then I tell myself, okay, maybe you can't read all that literature in due time, do some data mining instead, so using databases. Typically, the method is to obtain disease-associated genes. And again, if there is no disease-associated genes for immunotherapy resistance in melanoma, then you have to try merging different sources, and you get hairball, like the one you see below. And good luck finding context-relevant genes for the task you have been assigned. Then there's lunch break, typically. And after lunch break, I realized that maybe I could just use ENQUIRE, a software that reconstructs co-occurrences from literature and also expands. Now we will focus on the reconstruction part for this flash talk. So basically, I resume again for my fine-tuned literature that might give me more accuracy in what I'm actually looking for. And what the tool does in an integrative fashion for me is natural language processing and applies a graph-based statistics that you might not have heard of before for other topics of the same fashion to find not so random co-occurrences in your specific literature corpus given the entities that co-occur with each other. And the vanilla output that you get by default is a gene and mesh co-occurrence network, and we will use both of these entities to infer and characterize our case study of interest because we have available confidence metrics and topological clusterings to do further analysis with that. So, and the time is quite realistic, it's truly fast, and in about an hour, for example, I could try to annotate this gene mesh network that I obtained. This can be done, for example, with some embedding techniques such as node-to-vec. So now I basically have preliminary clusters assigned to my co-occurrence network. And if I employ fuzzy C-means, because I have now numerical values associated to all these entities, I can basically define custom gene sets that might help me, if I have available an omics data set, to fine-tune my GSEA specific to what I'm actually studying. And we use the mesh terms closest to these genes to characterize these gene sets. And you can always retrieve where they are coming from the literature. It's all reproducible and trackable. Another postdoc analysis that is already included in ENQUIRE is to combine it with a protein-protein interaction network such as STRING. Here we are talking about literature co-occurrences. And if we map it into physical interaction, we have a chance to do further community detection and see whether some pathways overlap. And then we get something like this, where some hubs are characterized and some are not. And these uncharacterized pathways can be further inspected if we use something like Neo4j, because ENQUIRE can be easily converted into a graph database representation. And then again, we have more informed research on the topic. So yes, about 6 p.m. I can pack, and I'm leaving with some data to analyze in the coming days. Next time it could be you. The paper, the ENQUIRE is published online, it's containerized, it's documented, it's benchmarked, and it's lightweight. You can run it even on your personal laptop. Thank you very much for the kind of attention, and see you at the poster session for text mining COSI and later on.
Thank you very much, Luca.